Hi guys, my name is Israel and I'm doing my presentation on the problem from the lesson 11, teach one another questions, number 2, which is the wobble rule question. And I'll just go ahead and start by reading it. It says this question concerns the hydrogen bonds that form between mRNA codons and tRNA codons, i.e. the wobble rules. And it's asking us, it says suppose an mRNA codon has a sequence 5' to UUU 3'. What are the sequences of the tRNA anticodons that recognize this 5' UUU 3' codon? So just kind of for review, I won't go too much into detail for time, but translation, what this is referring to, remember translation, it's when we translate the language of nucleotides or nucleic acids into the language of amino acids. And so we have our mRNA sequence, or messenger mRNA sequence, which would go into the ribosome, and we'd use tRNA, which is pictured right here, to match the code, which is in codons in pairs of threes, from the mRNA to an anticodon of tRNA in order to form a chain of amino acids, which would be right here. So each one of these three would code for a different amino acid, or sometimes the same one. And so you would typically just assume, if it's UUU, then it would match AAA. That's what we were used to, but the wobble rule tells us that there's something else that can happen. That something the cell uses or does to save energy, so that we don't have to use or create a bunch of tRNAs, and we can just use the same one. So what the wobble rule states, you can see this codon right here, is that the 5' most nucleotide and the middle nucleotide always follow the AUCG rule. So these two from the mRNA are always going to match the normal rule. We'll call it the normal rule on the anticodon in tRNA. However, the wobble position or the wobble rule says that the third nucleotide going from 5' to 3' on the mRNA can vary, can wobble. It's not stable. That's how I remember it. And so there's different ones, different anticodons that it could code for, that it could match up to. So we can actually just use this picture to answer our first question. So we're trying to know what different options we have for a UUU mRNA. So remember that if we're matching it up with our mRNA, we would start with the 3' end. And the first two that would match up are these UU, so that means we have AA because they follow the rule. However, the third one is the one that can vary. And so let's go to our chart, which is a chart that tells us which variations we can have of the tRNA. So what we do is we look at this third nucleotide and we find it right here, since we're going off the mRNA. All the places where it's at. So right here, right here, right here, and right here. There's 4 different ones. And so that means there's 4 different anticodons that can be used. So the first one would be it would end with G. So that's one of the answers. The second one, again, the first nucleotides would match up, so AA. And the second U is found at the A spot, so that's the third one. Our third answer, again, I hope you can see a pattern now. So the first two, AA, and then we find, again, another place right here where the U is, so it's at the U, so we can also have a U, 5'. And then the last one, AA again, because we're following the rule for the first two, and then U indicates that there can be an I as a tRNA anticodon 5'. So those are our 4 answers. I think when we do the second example, it'll become even more clear. So this says, suppose an mRNA codon has a sequence 5' GCG to 3', and what are the sequences of the tRNA anticodons that can recognize this codon? And so again, we would start with our 3' end, because we're writing a tRNA anticodon, it has to match the mRNA. And our first two would follow our rule. So we would have a G would match with the C, and a C would match with the G. So we know those first two won't change, and those are correct. Now the third nucleotide is the one that our wobble nucleotide. So in this case, it's a G. So what would you do? Since you're in the mRNA, then you would go to your chart and find in this column, third nucleotide of mRNA codon, all the places where a G is found. So we have 1, 2, 3. So that means there's 3 different tRNA anticodons. The first one is G to C, which is the normal one that we're used to, 5'. Our second one, again, we follow our pattern, so CG always is normal, and then G can also match with A, so we have that one. And then finally, our third one is CG, and then the other place where G sat is U. So that would be our third answer, and those are the 3 answers for part B. So I hope that part makes sense, because part C and D kind of switches a little bit. And what we'll do, I'll just go ahead and read it. It's basically the same, but it does have a little variation. You're given the T, suppose a tRNA anticodon has a sequence 5' IAA to 3'. What are the mRNA codon sequences that can be recognized by this 5' IAA 3'? So the interesting thing about this one is that since we're going from the anticodon, that means we have to work backwards. So our normal nucleotides would be A, and our third one would be the I, because we're working backwards. It's basically as if we were going from here to here. So our wobble position's right here. So the ones that would match, so 5' to would be, or the ones that would follow the normal rule would be these 2 AAs, which means that we would have UU. And then for the third nucleotide in the anticodon, we have an I, so we would work backwards. We would look at our nucleotide of tRNA anticodons and find I, and the 3 variations are U, C, and A. So those are our 3 answers. So U, and then again 5', UUU, because we have our first 2 that always match, and then C to 3'. And then 5' to, and yeah, UUU and A. And those would be the 3 answers for part C. And part D is exactly the same as that one. Essentially, we would do the same thing. So suppose the tRNA anticodon has a sequence 5', AUC 3'. What are the mRNA codon sequences that can be recognized by this anticodon? So we would work backwards. So C and U are matching up. The wobble position right here is A, because recall that the wobble position goes from the 5' and 5' and the middle one and the mRNA are the ones that are normal. So we would work backwards. So 5' C would match with G, and U would match with A. So those are normal. And then our wobble in this case is A. And if we find it in our chart, we can see that there's 3 variations, which are U, C, and G. So those would be our 3 answers. So U, 3 prime, and 5 prime. We have our normal ones, G, A, and C. And 5 prime, G, A, and G. 3 prime. And those are the answers. So I hope that was a good explanation of the wobble rule and how to do this problem. Thank you all for listening.